One sound clash. Good morning. Good morning. Good morning. Good morning. It's Friday. It's Friday. You did it. It's the end of the week. Gorgeous drawing. I agree with everything in the chat. That's pretty awesome. I cannot do this with my mouse. So well done. Great finding of the words. I hope that you have had a good week. I hope that you have been taking care of yourself and taking care of others, and that others have been taking care of you, because that's kind of the way this kind of thing goes. And welcome. This is the fourth of our installments, I believe, in the mollusks. We have one more. We're going to talk about scaphopods and bivalves on Monday. And today we're talking about cephalopods. I just glanced down at the chat and saw that some of you have a biochemistry midterm tomorrow. That's my official word on that. And I also see the question, Sophia, just wanted to confirm that the final exam is online in CourseLink. This is the case. Yes. In the coming days, I'll tell you, I can answer more questions, specific questions about the final exam. It is on CourseLink, and it's not going to be new. Right? This is kind of the point of doing the quizzes. I'm not going to switch formats in the end and have you got used to the way that I'm asking questions and why I'm asking the questions and how I'm asking the questions. And then all of a sudden be like, write me a thesis on this. It's going to be the same kind of formats. One of the things that I can tell you, I'm going to clear all these drawings. So say goodbye. Thank you for the octopus teacher. That was gorgeous. And one of the things I can tell you about the quizzes from now on is that we're going to transition them. There's kind of a before the midterm, after the midterm feel that in the before, there's some true or false. I'm not going to take away the true false, but I'm going to reduce the number of true false, increase the number of multiple choice and multi-select. Hooray. Alrighty. I'm still looking at the chat, so let me go down. Yes. Yeah, we'll talk about that a little bit. Awesome. Buckle up. Shall we begin? Where were we? Oh my God. So in this year, when we're doing kind of one asynchronous and two synchronous a week, it hurts my soul when there's, and I sometimes do move things around, when there's a whole bunch of things that I'm really excited about that appear in the asynchronous time, and I'm excitedly shouting to this computer in this room. And gastropods is really a time like that. So we talked about gastropods on Wednesday. We talked about thermal vent snails, snails made with iron, iron-born snails, snails that are reflecting light without a head when they don't have a head, still signaling to potential predators that giving them a dishonest signal of toxicity. We talked about snails that are leaves, snails that harvest algae, species-specific relationships between snails and algae so that they can basically be photoautotrophs. We talk about the foot and mucus and reproduction, and then we began talking about the cephalopods. And we left off talking about buoyancy, and that's where we're going to pick things up today. When we talk about cephalopods, this is kind of where the specifics of where we're going. We're going to talk about buoyancy, locomotion, all those things listed there. And these are all going to happen under the bigger picture learning outcomes of these three verbs, the appraising, the synthesizing, and the interpreting. Oh, the appraising, the synthesizing, and the interpreting that we're going to do in the context of thinking about how a mobile lifestyle has really shaped cephalopod evolution. We're going to be thinking about, in the context of those three verbs, about how predatory feeding lifestyle has affected cephalopod evolution and examples of cephalopod intelligence. So these three are clearly related, and so we're going to unpack that as if we were peeking out from a coconut on the bottom of the ocean floor. But I've got exciting news. Everybody put your hands up and go, exciting news. In today's issue of Science, there's a mollusk on the cover. And that's because they are announcing. Do you remember that dour phylogeny, the one that looked like basically my arm and coming up and then a polytomy of 7 or 8 things going, mollusks, we know they're real, but we do not know who's related to who. Well, and we've kind of unpacked some intermediate phylogenies along the way. Well, we have a well-supported molluscan phylogeny that came out in Science Today. And it actually, the senior author is the first author of the dour phylogeny, what I call the dour phylogeny, where it's the pessimistic one, where they look at it and they're like, we know nothing. They're like, and now they're like, oh yeah, we know some things. So we know that the polyplacophorans and the aplacophorans are close relatives, sister taxa. And then we know that there's a conchifera with an early branching monoplacophoran. And then we've got cephalopods, gastropods, scaphopods, and bivalves. There is one difference. I'll take away the pictures and put up the names, kind of rotate it. There is one difference to the phylogeny that we've talked about here in class. And it's pretty subtle, but it's the sister relationships here within the conchiferans, like as to who is most closely related to the bivalves. And this is the one that we've talked about, we've kind of used. Remember the triangles? Somebody had asked earlier about what did the triangles mean, and I was like, well, in this context, don't worry about it. It's just a way of me calling attention to you. And so it's this relationship here, so that now the hypothesis is that the scaphopods and the bivalves are the most closely related. They are a sister taxa. So this 3 still forms a monophyletic group, the gastro, the scaphopods, and the bivalves. But the sister taxa has been flipped. The specifics of why we're talking about that, it's not a thing that I would expect you to memorize. There's really nothing I expect you to memorize. I expect you to appraise, interpret, and synthesize. So why am I excited about this? Because it is literally today, hot off the presses, an example of the fact that phylogenies are hypotheses. And when we get better and new information, we revisit the hypotheses and the predictions we had thrown up with our old information, and we discard that which is no longer supported, and we move forward. And that's kind of amazing. So today, we're not going to think about biochemistry. We're going to think about cephalopods, because cephalopods are some incredible things. This was a picture that a former curator at the Royal Ontario Museum posted on social media a little while ago. So this is in Churchill. So while other folks are out looking around and standing bear guard looking for polar bears, he is looking at the skid of his helicopter. So for context, that's probably 6 to 8 feet long in between the frame of this bar at the top. And then you can see in the bottom here, this fossilized cephalopod. That's a big thing. Now, you've seen one of these kind of quickly in the past before, but this is in in BC. God, I just saw that scaphopod hermit crabs. That would be pretty threatening. These are so some other curled shell cephalopods. Now, on the tail end of our Wednesday time, when we had unpacked all that we were going to do about the gastropods, we talked about a little bit about buoyancy and how these septa, so these sections inside of extinct and extant shelled cephalopods, help them maintain buoyancy. So it lifts a big heavy shell that's protecting their body up off of the bottom and lets them kind of float and move through the water, which is kind of amazing. I'll unpack it just with a quick review to say the magic of it is by this connection line, this line that goes through the cephalopod, the siphuncle. And so these septa, so the animal lives only in the bottommost one. Okay, and then as it grows, its mantle secretes new sections of shell that it then seals off. But it maintains this connection via the siphuncle, and the epithelium, the skin lining of the siphuncle, this is a living kind of an organelle, maintains this high intracellular salt concentration. And so it's absorbing ions from the chimeral fluid, which is just the liquid that is initially in these different septa blocked off segments. And that creates an osmotic gradient. Water follows the gradient, okay? And it leaves the chamber, the newly sealed off chamber, and it enters the blood in the siphuncle and flows out into the body and then into the world. And as water is removed from the chamber, the gas that's dissolved in the blood diffuses into the chamber. And so therefore, these oldest chambers in the tight coils here, they're dry. The youngest ones out here, the large ones by the living chamber, they're moist and they could have like milliliters of water. But there are, this is how this big heavy shell, the shell that could be as tall as I am, is able to float and be a mobile predator and still protect the organism. right? That was previously on Zoology 2700. But as you know, because many of you, for this is in this class of the phylum Mollusca, reside animals that you are the most passionate about. What you know is that nautiloids, or extinct shelled cephalopods, they are not common anymore. There's a couple of lineages, right? There's several species of shelled nautiloids. Most are not. Most are octopus or squid. Squid's a polyphyletic term, but cuttlefish or squid. But these, where the shell is internalized and reduced to a small pen or really next to nothing at all. And so the octopus can actually just kind of, as you know, can kind of like sneak its way underneath doorways and that kind of thing. How do the extant squids do it? And it is kind of an amazing adaptation. It still involves heavy cations, heavy ions, heavy salts that are associated with their predatory diet. So here's predation. We talked about feeding lifestyle. We talked about mobility. We're going to mix the two of them right off the bat to say in the coelom, in the body cavity of the cuttlefish or the octopus, can be large reservoirs of fluid where they are slowly replaced by these low molecular weight ammonium ions. Ammonia is the toxic byproduct of a metabolism that's predatory, that's eating meat, right? And that results in neutral buoyancy. So the cephalopods, they're carnivores. Proteins are a major sort of their energy, and the byproduct, the end product is the ammonia, and that's released via the gill's inner epithelium into this container that they have that allows them to sit up in the water column and maintain this neutral buoyancy. And in fact, the rates of ammonia formation in squid, in some species of squid, are amongst the highest known among the invertebrates. That's just kind of sealing off the septa of thinking about buoyancy for either the shelled or the unshelled cephalopods. All right, I'm just scrolling through the chat here, and I see, it was pretty cool to see in nature to see the scaly foot Chrysomelum squamiferum there, the beloved octopus. Oh yeah. I am not from the East Coast. I am from Ontario. I'm from a region of Ontario called the Ottawa Valley. So on the Quebec border near, if you've been up towards Algonquin Park or whitewater rafting, that kind of area, that's where I am from. And proud to be. Now, once you're up off the bottom and you're buoyant, how do you move? Cephalopods do it in a whole bunch of different ways. They can move their muscles through their fins and have a fin. They can jet propulse, and they can walk. All right? Basically, if there's a way to do it, they can do it. So some of those are involving the mantle, the fins. Some of those are involving adaptations of the mantle that have been turned into this jet propulsion system, and some of those are involving the foot that has become these arms and tentacles. Let's talk first about the jet propulsion here. And we've taken a look at a cross-section here of a squid mantle. So we've got the pen at the top, that little dot hanging above it. That's the remains of the internalized shell, right? That's providing some rigidity. And take a look at all of the rings of circular muscles. So there are some radial muscles that are running back and forth, but then circular muscles that are running around that cavity of the mantle. So what we've got is, on the left-hand side, the idea that there is an exhalant phase. So just as you've seen in so many other invertebrate organisms with a hydrostatic skeleton, we see, boom, the circular muscle squeezes and there's a displacement. In this case, it's not the coelom. It's not the body, like the internalized liquid of the body that's moving around. It's, in fact, water that's being shot out forward. And then when it inhales, and these radial muscles fire and it inhales, that sucks more water in, so it can go boom and shoot it forward again. So when it's moving slowly, the circular mantle contracts, and then when it's moving fast, there's a rapid contraction. Okay, so the fast jetting, the funnel can actually be changed. So the funnel or the siphon can be pointed anteriorly or posteriorly. So there's a movement around that can be done. Let's take a look at a different picture for that. So we've got the circular and the radial muscles. And then we've got arms first swimming, where the funnel is pointing. So basically, that's kind of like predatory, right? That's kind of moving around, thinking about, looking at what it's eating or what it's potentially going to eat. Now, if it needs in tail first swimming to escape, it can flip around and flip the funnel around and boom, shoot backwards. And you've seen videos of these kinds of things. And I will show you more of them. So this is a gigantic squid off of the Pacific coast that is using, let's take a look. You're seeing some of the mantle of the fin swimming here, okay? So the fins are muscularized as well, and so once the organism is sitting in the water column, if it's not jetting around forwards or backwards, it can move itself very slowly. And one of the things that happens that we can unpack further with time in 3700, if you take 3700 next year, is the fact that as squid age, they change. This is, I'm going to say something incredibly obvious, they change in size. As they change in size, how they swim changes as well. Okay? How they orient, how they're buoyant, how they maintain buoyancy, and how they move changes as well. So, what about the walking? So we'll think about octopoids here. And as they're changing in color, which we'll talk about in a second, they can pick themselves up and say, I'm out. It's a slow burn, this video, right? Like you're watching it going, okay, so they're kind of moving on their tentacles. Doesn't really look like walking. Until in the end, it looks like it's hiking up its kind of Victorian skirts and running away across the bottom of the ocean. I glanced down at the chat, and so, Zara, that's exactly what I was, yes. So their movement does depend on the surface shape and the size, and it changes. And in this class, this year, we're not going to have a lot of time to unpack that, and that's kind of what I was nodding to and saying, oh, take 3700, and we'll talk more about how these things happen.
You want to get up and get the ladder?
Get ready for it.
As you can see, some children in this aquarium in Seattle were not entirely pleased with the octopus deciding that it's going to get out of its container and walk away. And then there's another one of from the Daily Mail a few years ago about this, a cheeky cephalopod snatches the camera and takes a picture. Yeah, it's a great question. I wonder what they think of all the people. I'm guessing they do think something. So they are up off the bottom. They're buoyant. They can move with their fins. They can move jet propulsed. They can walk. And they're going to be predatory. We've kind of stated that, right? So one of the things about that is a real change. The beak and the radula become this raptoral kind of a harvesting organism. So we've got a radula still inside, but we've got jaws, raptoral jaws. Literally, like if you've seen the beak of a raptor, of a hawk or a falcon, tell me that does not look. I mean, we talk often about cephalopods in convergent evolution when we think about the camera eye with our vertebrate eye and their eye and think, wow, this is incredible. If you're going to see this is really how to do it. We should also be talking about if you're going to be a raptoral feeder, you need that kind of a beak. That is a really great way to walk around harvesting and eating things. So in and amongst this, we've sort of got the radula. There are also these posterior salivary glands that secrete proteolytics. So they will actually help enzymes that help to digest, to anticipate the digestion and to facilitate rather the digestion and to speed it up of these protein of the animal tissues that they are eating. So remember, we've talked about raptors, we've said raptoral, we said they're predators. This is a departure. The ancestral mollusk, this monoplacophorine looking thing that's rasping the trails that those excited nerds on the remotely operated vehicle, the ship operating the remotely operated vehicle were shouting, monoplaque, monoplaque, monoplaque. This is such a departure from that, right? This is a time I'm asking to reflect on the fact that this is one phylum that has forms so vast and divergent. I mean, we're going to get to the arthropods. I have a special warm place in my heart for the arthropods, but the variety of forms and lifestyles that we see within the mollusks gets me up in the morning and makes me excited about life, right? A camera eye here. A mantle that's seeing and evolves into an eye in the bivalves, and then the shell that evolves into the eye in the polyplacophorines. That's bananas. So also involved in this part of, if we take a look at the alimentary canal starting with the mouth, we see the ink sac. So this is a diverticulum of the rectal wall, and the ink sac has muscular walls that can inject ink into the exhalant water. So the water that's not excellent water, but the exhalant water that's coming out. And that's what the video is kind of looping through here, is showing as it kind of moves away that there are melanin granules. But interestingly, there's not just melanin granules, there's a whole bunch of mucus. And the mucus, one of the hypotheses is, and we watch it in slow-mo, boom, it belches it out. That helps the melanin granules suspend in the water column, holds them there so they don't fall out or into the current of the water column that might be there. And in some cases, and this might be some hand-wavy human anthropomorphizing, but we think one of the ideas behind the suspension of melanin in granules in mucus is that it hangs there and essentially forms this shape. Potentially rapidly interpreted as a large predator of whoever was threatening predation upon the cephalopod. And so the idea is that much in the same way that the eye spots on a moth or on a caterpillar, really they only have to be good enough to make a predator second guess for a second. The same kind of thinking goes into the mucus and the melanin. All right. So thinking about cephalopods in a macroevolutionary sense, from like extinct forms to very extant and affected by the climate crisis ways. There on the west, this is wrong, it should say Western North America. Dun, 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 dun. And that bugs me, so I'm going to change it. So Western North America, the Humboldt squid, this giant squid, is its range, which historically has been very biased towards the equator and towards the southern parts of the southern hemisphere. It's moving north, and as it's it's a big predator, right? It's meter and a half long. This is a great big animal, and its range expansion is coinciding with climate associated changes in oceanographic conditions, and therefore it's associated with big changes in other competing top predators. So fisheries on the west coast off of California, Washington, British Columbia, this is a thing that's coming. This is a thing, and we think, okay, well, maybe that doesn't affect me. It really does. Even there's going to be an intersection historically in your lifetime, likely between the Humboldt squid and the salmon fishery that never would have bumped into each other in the past. They eat, what do cephalopods eat? All the things: fish, crustaceans, other cephalopod, benthic invertebrates, and I'll park it there. Another statement about 3700, what they eat changes dramatically as they age, because they go from being we've talked about kind of thrown out the Reynolds number. They go from being a very smaller than a millimeter to a meter, so like orders of magnitude change in their body size. And so, of course, they're affected by the world very differently. And then, of course, they eat very different things. Now, let me pause this for a second. What do they eat? This is another, this is an expression, an Australian flavored expression of delight or terror, watching the fact that some of these cephalopods, because they can walk and because they can deal with being out of water for, as you saw in that aquarium video for a little while, sometimes predate over land-ish in tidal pools. I hope you can hear some of the audio here. There's the cephalopod. There's the crab.
Shuddy! There's an octopus eating a crab!
Bye-bye, crab.
Holy shit!
And of course it has to finish with that common statement from Australians. Yeah. All right. Where were we? There we are. All right, that's a reminder for Caramel to tell me where I am. That's amazing. Okay. All right. We'll keep going. We're going to talk a little about, remember we're thinking about these in the learning outcomes, the bigger picture kind of narratives or stories within the cephalopods as we dive into some taxonomic and specific examples is about predation, mobility, intelligence. And so one of those then has to do with the fact with their hemal system, right? Their gas exchange and their excretory system. So flow here is very different than that ancestral mollusk. This is a closed system. One of the things that characterizes cephalopods compared to other mollusks is that they have a closed system, circulatory system with tissue and capillaries. So capillaries that get right into. So this is not a circulatory system that bathes the organs and the tissues in a liquid that contains a respiratory pigment. This is like just like we have, like tissues and capillaries that get right into and do the business. All right. So they have bipectinate gills. That's what you're looking at here, these little honeycombs. And the hypothesis is that third bullet says is that this closed system is an adaptation to support this buoyant predatory lifestyle. So the capillaries provide this greater array of contact between the hemal system than would be provided in large sinuses. That's what I was saying when the organs or the tissues are just bathed. There's none of that intimate kind of capillary connection. So we've got a systemic heart that is pumping out and then back through into the bipectinate gills, one branch through, boom, out again. So very kind of grossly similar to what you would see if you modeled our own. Here's another drawing from your text. We're in chapter 13. Remember the giant chapter 13? So you can see the branches of the bipectinate gills and the drawings slightly different, but really just showing the same things, the same elements. And I've got another text that is comparing and contrasting a closed and an open system from two different mollusks. And this is a shout out to next week, because this is on Monday when we'll be talking about bivalves. This is a bivalve system. Okay, so in an open system, the vessels, the blood vessels empty into the gonocele or the coelom, basically these sinal cavities, these sinusoidal cavities, which make up a big part of the body volume in the bivalve that's in between its valves. All right, so there's a lower pressure, lower blood pressure, limited opportunities to change the velocity, to alter it, and to change and affect rates of blood flow, rates of oxygen transfer. Whereas in the closed system here that's modeled in, now you're familiar with, in the cephalopod form, we have higher blood pressure, which is a good thing in this case, and a particular ability to alter velocity in the distribution of blood. And the total volume of the hemocoel is much reduced. It's only about 5 to 10% instead of 15 to 30%. And the real functional difference is because of that surface area that the capillaries allow the transfer directly. So bringing the oxygen directly to the system, to the tissues that are doing the work. Now, this is connected in the cephalopod world, in the mollusk world, really, to excretion. We talked about them already as being excreting soluble ammonia, so ammonotelic. I'm just going to run at that. So, and we talked about it in terms of squid, cuttlefish, kind of maintaining that neutral buoyancy. They have this capacity to excrete soluble ammonia because they have this urine, this renal sac that is essentially going to create and collect urine and provide with these fingers of these kind of, see the renal appendages kind of hanging down into the renal sac. This is providing as the blood is flowing in and out through the renal sac and then through the renal appendages. As they intersect there, that surface area is allowing the selective reabsorption of different compounds that they don't want to excrete. In this case, maybe the retention of ammonia. But then ultimately, through the nephritopore, they are excreting urine out into the mantle cavity that will be excreted or moved out into the water as they move through the world. Now, the nervous system, we're going to unpack for a bit of time now, is strongly concentrated, strongly cephalized, and bilaterally symmetrical. This is how we get into thinking about intelligence, because there is extreme cephalization and giant motor axons. So we've got these first, second, and third order giant cells that proceed from anterior to posterior of the body. They're capable of and very innervated parts of the body. So you can see the fins here on the side that can undulate and move them through the water versus the innervation of the mantle itself that is going to fire and allow them to escape, and then not diagrammed here, but of course innervation into the tentacles that are allowing them to capture or to explore the world. Now, before we get too far, I want to check again. I've arranged for Caramel to show up here, and I don't see... Yeah, everything's all fun and games until an Aussie gets scared. All right, we're going to pick up our thinking about intelligence, and we're going to think about it in terms of tool design and tool use. So 15 years ago, roughly 15 years ago, there was a paper published that documented defensive tool use in a coconut-carrying octopus. So this was an octopus, and many of them, this was not just a single observation, but this was an observation of the frequent observation of octopus using discarded elements on the bottom that they would find in the bottom of the ocean to essentially hide in and hide under. But more so than just kind of occasionally running by something and then ducking behind to hide. This is the decision of the cephalopod to maintain and to keep contact with that cover item and to bring it along. To apparently have judged the efficacy of this strategy and thought, oh, this is what I'm going to need to do this again. So this is clearly tool use, which, and you've bumped into, I'm sure, this in your training in vertebrate zoology and just in your broader training as being a biologist. We often have this bias in our own species to think about tool use as being something that really characterizes our own species. So not just of vertebrates, but in fact just of us. When in fact, there's a great view of the siphon there right beside its eye as she's peeking out. One of the things that we learn is we learn more and more about there's a lovely view. I love it. One of the things that we learn is we learn more and more about the species that share their planet with us, is that tool use is certainly special, but it's not a thing that characterizes us. That there are many taxa, including cephalopods or octopoids, that do it. And so this causes us to rethink how we think about it, as opposed to being like a saltatory thing that occurs in humans. This is maybe a continuum of tool use, from insects to primates, that provide an opportunity for us to rethink rethink the othering that we do with other species all the time. An opportunity for us, particularly those of us lucky enough to train ourselves as biologists, to be informed by by other species of our own biases. Now, one of the cool studies that's come out recently with regard to tool use in and around the same area that the coconut shell was used. Now, you can see just by that cut, this is garbage, right? This is a type of marine. This is a cut that has been done with a machete. So this is human waste that is in the water that they are now using. And there's a bunch of studies that have been done to look at, in this part of the world, octopus and other cephalopods that are intentionally using garbage, marine garbage, as cover items. So, how did octopuses get so smart? So one of the idea, because the cephalopods, the intelligence we see in cephalopods challenges kind of most accepted hypotheses about the evolution of intelligence. Most of those have to do with living a long life and raising a small number of young. And so we think this is a bias in terms of, well, we are intelligent to a greater or lesser degree. And so this is how we see our own evolutionary trajectory, is that it became more and more longer lived and based around the cultural transmission and the group-based transmission of things. Now, cephalopods are not any of those things. They're not group-based generally, and they're not long-lived. But they are clearly intelligent. So they have a complex brain and a behavioral flexibility in a simple social environment and with a fast life history. And so complex social environment and a slow life history were really what we thought were the mechanisms that would result through evolutionary time in intelligence. So it's kind of surprising from an evolutionary perspective that cephalopod intelligence has been overlooked. And so this leaves this evolutionary conundrum kind of unresolved. Now, many of you have seen my octopus teacher. So there's all sorts of examples in the literature about cephalopod intelligence. I'll provide you one here, which is researchers so kind of go from left to right in these three different. So there's cephalopods that are put inside of an environment that has a background that is speckled and a background that's lined. And they have, there's no poking or prodding. There's no pain. There is going to be pain in this. So there's a noxious stimulant. So then there's a treatment where there's a noxious stimulant applied in the stripy area. And what the cephalopod does, what the octopus does, is conditioned to avoid that place. Then in a third treatment, the same animals that have been exposed to the second treatment are placed in the same kind of background environment, but with no noxious stimulus. And what they do is they still avoid the area that they had received the noxious treatment in the past. So this is it's a very simple expression of intelligence. So what do I mean by noxious? I mean something that hurts them, something that's uncomfortable. Now, I can tell you that to do this experiment, they would have had to go through the same kind of animal care protocols that to do any kind of an experiment in a university setting with vertebrates. This is the one invertebrate group where you need to go through the same animal care protocols to demonstrate that if you're going to cause pain or use a noxious substance, that it's been evaluated by your peers as being worthwhile. If I decided to do the same thing with beetles, I do not have to do that. And we can have a long debate about, or a discussion about why that might be. But the cephalopods are the one invertebrate group for which you have to apply and use the same kind of animal care protocols that you do in a vertebrate. So they avoid a location after this very simple demonstration. So octopus holds a grudge. You know, there's a beer, I think, Great Lakes Brewery? The octopus wants to fight. And that's because octopus holds a grudge, because octopus remembers. So this is the noxious substance, the dilute acetic acid. And the bottom is talking about their branchial connectivity, so high activity in their nervous system. And so they're capable of both discriminative and effective pain experience, and that's the difference between kind of 2 and 3. Taylor, I just glanced down to see your question. What's the line for that? Is that public consensus, or is there a specific criteria? What's that referring to? Oh, are you asking about the animal care protocol? Yeah, so it has to do with our increased appreciation that these are very intelligent organisms. And so if they're very intelligent and we can design experiments where clearly they feel pain and clearly they remember it, so we can then associate it with negative feelings, with feelings themselves, there's still a large debate in a lot of insect taxa or invertebrate taxa, rather, as to whether or not they feel at all. I think probably we can unpack again. Maybe I'll design a lecture to think about this, but the almost certainly they do. And so my guess is that if you get to replace me in this career and you get to this stage in your career, probably the number of invertebrate taxa that have the treatments that vertebrate-like treatments that cephalopods do will increase. How long do they live? Does their intelligence decrease or increase as they age? It's a great question. So they do not live long, a season, right? So there's not a whole bunch of time to accumulate experience. But there is, if they are clearly intelligent. So my guess is that they are fast learners and that they would become more intelligent, more wise as the year goes on. Why are they so intelligent? Why? And we think it has a lot to do, and this is linking back to this, thinking about buoyancy at the start. There's kind of two hypotheses, and one of them is they both kind of link back to this idea that in losing the shell, that was a big traumatic event. The shell, seen here in the extant nautiloids, this is a real protective device, right? Ask any snail, except for a slug. The shell is a real important way to prevent the world from trying to eat you. As soon as you lose it, as you internalize it and become as soft as an octopoid, there's a dramatic increase in what can eat you. If you're soft and gelatinous, most things will eat you. Most things will try and eat you. So the idea is that this prevents an increase, so the loss of the shell, increase in the rate of predation, and that essentially prevents the emergence of a slow life history strategy. What's a slow life? Like long-lived, like a bivalve that can live for 500 years, for example. And there are such bivalves. There are mussels that can do that. We do not see that in the cephalopods because bivalves have the shell. They can hunker down. There's no hunkering down and waiting something out that a cephalopod can do. But losing the shell also allowed new niches and new exploitation of novel environments. And so then the hypothesis is that this combination of escaping predation, exploring new niches, becoming predatory yourself, this is what then favored a fast life history strategy with intelligence. I'm just looking and I see Kayla's question. I always thought that octopus lived long. Aren't there old octopus in aquariums, or is it just that they don't live long in the wild? So they do not live long in the wild, even in the aquarium. So certainly in an artificial environment like an aquarium, they will live longer than they would in the wild because we're controlling predation, right? But you would never have an aquarium their life history. They burn out. They will die. They will not live decades into hundreds of years as a slow metabolized like a bivalve would. Yeah, and there's some other examples in terms of reproductive strategy. How long do nautiloids live? It's a great question, and I don't have a number off the top of my head, but it'll be longer, and it won't be a year. It'll be multiple years. It could be decades, even, is my guess. But if you look that up, I'd love to know what the answer is. Now, thinking again about intelligence and about eyes, this is the last kind of area we're going to talk about for the last 5 minutes here. And because complex image forming eye, right? Like this is not a cross section of our eye, even though it really almost could be, is that we've got this iris and the cornea and then the retina and the optic nerves. This is a cross section of a cephalopod eye. This is a real classic example of convergent evolution, that if you want to see the world, remember we talked about our bias? It's kind of a shared bias here when we use to see as to understand. So this is an example of that complex camera eye. So there are other ways, the other sense organs that they have. They have otic capsules, which are statists on either side of the brain that detect gravity and acceleration and turning. Some of them have a lateral line or multiple lateral lines. That's what you're seeing here. Epidermal lines to detect vibrations, and they can smell. They are not just in a way that they're better at this than we are. They are both visual and they are smellers. So they have olfactory organs. Now, one of the really cool things to me, and I'm sure to you, about the cephalopod is their capacity, their chromatophores, their capacity to change color. Like that flamboyant cuttlefish, that's ridiculous. And there it is. So they have multiple organs, multicellular organs that are associated with their central nervous system and a central pigment cell and radial cells that can inflate and contract and expose light reflecting cells. They have, and I'll show you a diagram of that in a second. So the contraction of these radial muscles dilates this pigment sac and converts it into a large flat plate. They also have iridocytes, which are a structural generation of color, and photophores, which can be housing for bioluminescent bacteria, which we'll talk. So we'll talk about all these. So here's this disc. So you see that at the bottom, this oval that has the rays sticking out of it. Those are the muscles that are going to be used by the cephalopod
cell is
to change its color. And this is a little diagram from PBS NOVA to show that.
this little ball of color, all tightly bound up so you don't see it, and it has muscles attached to it, and the muscles can pull that pigment sac out into a little disc of color. And then when the muscles let go, it just boink, goes right back in and you don't see anything. And so it's a very simple mechanism.
Very simple mechanism and
Next,
super
there's
elegant.
a deeper layer of
Okay, and gorgeous. So here's some of baby squid that when they bumped, this is again, so they're going to slow the video down. And you can see these tiny little iridophores, and then boom, they flare out. This is zooming in and setting them to music. But these are changes that are associated that they can do in real time. Remember we talked about how highly innervated their body is, how cephalized. And we see these incredible changes in shape and form and color so that there are, if we leave them on a checkerboard, they will begin to look like a checkerboard. If we leave them in a coral reef, they will begin to look like a coral reef. And then there's the mimic
Unknown
octopus.
until it was spotted, first by fishermen off the coast of Indonesia in the 1990s. It looked like an octopus, but it could morph its shape in an instant to appear as seemingly any animal around it. At first, no one had any idea what it was.
The first time I saw it, I just was blown away. You couldn't get a more spectacular animal. It really is the pinnacle of wizardry.
The pinnacle of wizardry. So we've got, I showed this to my boys last night, and they had properly a what? Wow, kind of to all of these different changes. Let's go back. I like the narration on this one.
Mimic scuttling along the sea bottom, looking something like a furry turkey with human legs.
Something like a furry turkey with human legs.
I have to admit I was screaming when I got this video thing.
What makes a marine biologist scream?
That's the one with Blue Jack's mic. Yeah. So we go through, and I think if I remember correctly, I think the eye is here. Yeah, no, I was even wrong then. That's amazing. Just amazing. Now, one of the things in the last couple of minutes I'd like you to do, throw me in the chat or on the screen. We need some hypotheses because they're incredible mimics. They're incredible camouflagers. They use color. They are themselves colorblind. Perhaps you know, or perhaps you have some hypotheses. Find some, either annotate the screen or show me in the chat why and how. And I'll give you some examples. Or I have, that's great. Something to do with their squiggly eyes. Yes. Their mimicry ability wouldn't compensate for the lack of shell. So why the life history? So that's an interesting hypothesis. So why doesn't mimicry compensate directly on a one-to-one with the loss of the shell? And apparently it doesn't. It's good, but it's not good enough. So it's better to have a shell than to be able to camouflage yourself as practically anything. Chemical sensors, poor eyesight, squiggly eyes. Yes, in a nutshell. One of the hypotheses is actually the eye. And the other is they might be seeing with their skin. They might be seeing color with their skin because they have photosensitive opsins, these pigments, in their skin. And the other is that they might be using what's called chromatic aberrations, so the differential bending of lights, of wavelengths, through these oddly shaped pupils, oddly to us, differently shaped pupils. So this might allow them to detect color by monitoring image blurring as focus changes. As you might imagine, both of these things are very interesting to biology-informed engineers, and so there are a whole bunch of studies that you can unpack to look at the dynamic camouflage that can be used. Now, last kind of example here about light is that there are squid that have these pockets where they keep bacteria that can bioluminesce. And so not only can they make their own colors, they can they make houses for things that can then make color for them. And in fact, we think that some of these giant squid are actually using these in a way to provide beneficial camouflage from predators and above, and signal to each other. And that's what this critter cam is showing, that they think these are signals, like almost like a semaphore that's being used by the squid to talk to each other. Kind of amazing. Okay, so I've gone about a minute over, my apologies, but cephalopod's kind of amazing. We've been here and we've talked about these things, and if you haven't seen it already, even if you have seen it, take some tissue and go and watch it again. It's gorgeous. So this is it. 2 minutes over. My apologies. You got places to go. Be good to yourself. This weekend, do something that's good for you. Be good to other people around you. And I'll turn off the recording in a second and then hang around if there's any questions. But well done you. You got to the end of the week.